Hi, this is Group 2. Our topic is disease cohort analysis. We have 4 people in our group, thus Rachel, Alanda, and Shah, which is me. This is the outline of the presentation. I will make a brief introduction of the project. Then, Alanda will talk about the experience of using CLC Workbench and ANOVA. Seth will show how we worked with VAST and Fever. And finally, Rachel will talk about the genetic test report we generated. The whole idea of this project is using different tools to find out the genes that are responsible for the given phenotype within a cohort. This is the phenotype description. First, the disease is autosomal dominant inheritance. That is, the abnormal gene is located on one of the 22 non-sexy chromosomes, and the child can be affected if any of the parents have the gene. The individual have this gene usually develop hundreds to thousands of colon and rectal polyps. Some of the polyps may lead to cancer if not surgically treated. We're also given a VCF file. This figure shows how the VCF is generated, and it can be divided to 3 parts. First, data cleanup, then variant discovery, and finally evaluation. By doing this, SNPs, indels, and structural variation codes can be presented. The VCF file we got was created by randomly selecting 10 Caucasian individuals from 1,000 genome exomal dataset. It represents the full exomal sequence data for the affected case. In all, our objective is to discover the gene underlying the genetic condition affecting these 10 individuals.
Cohort analysis. For cohort analysis, we chose 4 different software packages available. First one being CLC Cancer Workbench, second one ANOVA, third one VAST, and then Fever. The VAST results were actually input for the Fever analysis. Moving on to the next one, that is CLC Cancer Workbench. It has been designed for cancer-focused informatics solutions. For this, the first step which we followed was import the VCF file with tracks. Second step, that is compare shared variants in case versus control groups, for which we found certain limitations. The first one, and the foremost important one, was software has no provision to generate a graphical plot for analyzed data. Next one is ANOVA. ANOVA is specifically designed for functionally annotate genetic variants detected from diverse genomes. It performs gene-based annotation, region-based annotation, filter-based annotation, and has other functionality. From our analysis, we found out there are certain limitations for using ANOVA. The first one and the foremost is it is limited to variant from single VCF file, and the second one is APC was not prioritized in our result.
Hello, this is Seth Russell. I'll be talking about the tools we were able to successfully use to generate the information necessary for the genetic test report. While we have talked briefly about VAST in class, I think it would be helpful to start with a review of what VAST is and what it does. VAST is a command line tool written mostly in Perl and is the result of a collaboration between the Yandell Lab at the University of Utah, the Huff Lab at MD Anderson Cancer Center, and Omika Inc. VAST is a probabilistic search tool for identifying damaged genes and their disease-causing variants in personal genome sequences. The purpose in using the VAST tool is to search for regions of statistically significant dissimilarity due to sequence variants between the target and background genomes. VAST combines variant frequency data with amino acid substitution information on a feature-by-feature basis. VAST uses the likelihood ratio as described in Yandell et al. 2011 genome research to search for damaged genes by comparing the variants in a set of diseased genome cases to those in a set of healthy genome controls. Here's a high-level graphic showing the workflow for using VAST. The inputs to VAST are 1. Set of target or case variant files in either VCF or GVF format. 2. A set of background or control variant files in either VCF or GVF format. 3. A set of features to be scored, usually genes, and this should be in GFF3 format. 4th, a multi-FASTA file of the reference genome. Provided with these 4 inputs, most VAST analyses share these three basic steps in common. 1. Variant annotation. The variants for the individual genomes are annotated for the effects that they cause on the genomic features. 2. Variant selection. Some set of, or commonly all, the variants in a target genome are combined together for comparison against the set of all background variants. 3. Variant analysis. All the variants found in the target genomes are then compared to all the variants in the background genomes, and the features that contain those variants are scored and ranked by the likelihood that they are disease-causing. Here are some of the specific steps we followed to actually implement the workflow shown on the previous slides. We ran VAST on the CHPC Ember cluster. As a warning, VAST takes a bit of work to get going due to multiple Perl versions, as well as some missing modules and libraries. So as the first step, we converted the VCF file to GVF format. We then sorted the contents of the file as required by VAST. Next, we ran the VAST annotation tool on each of our GVF files. I've shown only one here, but we did it for all 10 of our sample cases. Next, we indexed each of the files and then merged them together into one condenser file. Once we had our CDR file, we ran several different iterations of VAST using different command line options in an attempt to find the best candidate genes. Lastly, we used the VAST Imager tool to create a nice Manhattan plot of the results, which I'll talk more about next. Here are some of the Manhattan plots we generated showing a representation of the various candidate genes and their significance. The key difference to note here is that with successive permutations over the data, VAST is able to narrow down the negative log base 10 of the p-value. When moving from 10,000 permutations as shown in the top image to 1 billion permutations as shown in the bottom image, the candidate genes stand out significantly from the background genes. Now that we've run VAST, the next question is how to determine which of these genes, either in the list or others, are the cause of the actual cohort disease. Here are some of the top gene results. While I've shown just the top 5 from the various runs of VAST, there are over 19,000 results provided from each run. Of course, not all results are statistically significant, but there are still many different results that need to be evaluated and reviewed to further narrow down the field of candidate genes. The next step we followed was to plug the candidate genes into the various tools such as NCBI and GeneCard. Here are some details on the top genes from GeneCard. At the top, PAX5 does seem to have a correlation with cancers, but not of the type from our cohort description as reviewed by Shaw. The APC gene does appear to match the cohort disease description better, but the question still remains, which genes are the best fit? To solve this problem, we turn to Fever. This application is a web-based tool that takes VAST output and uses information in gene ontologies to better prioritize and filter results. Specifically, Fever integrates phenotype, gene function, and disease information with personal and genomic data for improved power to identify disease-causing alleles. Fever works by combining knowledge resident in multiple biomedical ontologies with outputs of variant prioritization tools. It does so using an algorithm that propagates information across and between ontologies. This process enables Fever to accurately reprioritize potentially damaging alleles identified by variant prioritization tools in light of gene function, disease, and phenotype knowledge. As the key initial input to Fever, we had to come up with gene ontology terms from the cohort description, and those brief phrases are shown here, including polyps, desmoid tumors, and other nasty colon and rectum issues. So here are 3 basic steps to running Fever. I already mentioned the input to step 1, but I've included a screenshot here to show how it works. Step 2 was to provide the VAST output and VCF file. Step 3 is to configure Fever to determine how items will be prioritized or filtered out. The end result is this screen shown here, a prioritized list of the candidate genes with ranking. Clearly, APC is our winner. So now that we have APC as the winning gene, We will next turn to Rachel, who will describe the genetic test report as well as more details about APC mutations.
So this is Rachel here, and my part of the project was to take all the data that we generated using VAST and Fever and create a mock genetic test report. Shown here is the test report itself. Don't worry too much about reading this. I'll show close-ups of each section in the upcoming slides. I generated the test report using our data and followed a template published by the ACMG Clinical Laboratory Standards. The first section of our mock test report contains the information for our hypothetical testing laboratory, again borrowed from the ACMG template. This particular template is designed for genetic testing of a single individual, so I had to adapt it a little bit to fit our cohort of 10 individuals. In general, this section lists information like the patient's name, date of birth, pedigree number that that individual belongs to, the gender and race of that individual, as well as information about what the type of sample specimen they obtained as well as when it was obtained. Most of this data is extraneous for us as we did not directly sample these individuals, but it gives you the general idea of what one might see in such a report. The values that are relevant to us include the number of individuals, in this case 10 in the cohort, the number of males and females, 4 and 6 respectively, and the race of these individuals, which is white. Shown below the header is the test we performed, in this case exome sequencing, as well as the reason or indication for the test. As Sean mentioned, all individuals presented with at least one of these symptoms: adenomous colonic polyposis, desmoid tumors, osteomas, neoplasm of the rectum and colon, and colorectal cancer. We were also told that it was an autosomal dominant disorder. Thus, this section of the report outlines the goal of the test, which was namely to identify which gene is damaged or mutated in all the cohort individuals and leads to these shared observed symptoms. The next section of the report details the results of the test, indicating that it was either success or positive, as was the case with our test, or negative. Below that is a list of the variants of interest, including the gene that they belong to, the disease that gene is associated with, if any, the HGVS standardized classification or listing of these variants, as well as their clinical significance, if any. Using our methodology, we identified several variants that were likely pathogenic, and all of them were mutations in the APC gene, which was the top gene prioritized by Fever, based on our inputted list of GO terms. Most of these were deletion mutations of at least either 1, 2, 4, or 5 nucleotides in the mRNA sequence. Note here that instead of mRNA coordinates for these deletion variants, I only list the genomic coordinates, because the reason for this is because VAST outputs only the genomic coordinates. It doesn't output the coordinates of the mutation in the mRNA sequence, and I had a hard time reliably lining those up with specific changes in the mRNA sequence, so I've only listed the genomic coordinates. However, because this is exome sequencing, all of these deletions occur within the coding region of the protein, and because they are deletions of nucleotides not in multiples of 3, it's likely to lead to frame shift mutations, resulting in either truncated protein products or full-length protein products with scrambled amino acids. Thus, based on this evidence and the known effective mutations in the APC gene, which I'll discuss in the next slide, we classified these as being likely pathogenic. In addition to the deletion mutations, there were also 2 missense mutations in the mRNA, resulting in a valine to aspartic acid change. We classified these as uncertain significance because when we tried to verify their pathogenicity using the polyphen-2 and SIFT prediction algorithms, we got conflicting results. Polyphen-2 predicted that they would likely be benign mutations, whereas SIFT predicted they would not be tolerated changes. As such, these could potentially be pathogenic, but we just can't say for sure. The next section of the test report is the interpretation summary of the variants. It outlines the potential function of each variance, or the variance as a whole in detail, including what is known about the variant, the gene it is in, and what disease it may be causing. So what is APC, the top gene in the fever candidate list, and that has all these mutations in the target cases, but not the controls? Well, APC stands for adenomatous polyposis coli, and it is a tumor suppressor that inhibits the Wnt signaling pathway, which is involved in cell proliferation and development. It's also known that over 700 mutations in APC have been associated with an autosomal dominant disorder that predisposes individuals to colorectal cancer, especially early in life. And the name of this disease is familial adenomatous polyposis, or FAP. And this was really exciting to us because when we researched FAP further, we found that it closely matched the case description that we were given for the individuals. They told us it was an autosomal dominant disease, which FAP is. Individuals with FAP develop thousands of benign polyps in the colon, some of which will become malignant. This leads to an average age of colorectal cancer onset at 39 years in these individuals, and they also exhibit growth of fibrous desmoid tumors throughout the body. Because these symptoms match nicely to our observed symptoms in the cohort, this lends credence to the possibility that these mutations that VAST and fever identified in APC are the true causative disease-causing mutations of these individuals' symptoms. In addition to FAP symptoms being derived from APC mutations, it's also known that nearly any mutation in the APC gene will lead to colorectal cancer. The reason for this, additionally, APC defects are most severe when they are due to a truncated protein. The reason for this, which is likely the result of our deletion mutations, as I mentioned, and leading to frame shifts and truncation of the protein. Based on this evidence, we conclude that APC is the damaged gene responsible for the cohort symptoms. The final section of the test report contains our recommendations for treatment and response, as well as the potential limitations of the test. In our case, if we're practicing physicians, we would likely recommend that individuals in the cohort undergo genetic counseling and that all their offspring should be screened for heritable mutations in the APC gene. We would also recommend consideration of prophylactic removal of the colon because the incidence of colon cancer is very high in individuals with FAP and individuals with APC mutations in general. There were also a few limitations to our general test and experimental design. First, the cohort we were given was relatively small. Additionally, the software we used to verify the pathogenicity of some of the variants, such as polyphen-2 and SIFT, cannot be used for deletion mutations, which is our largest category of mutations that we identified. And finally, because we only utilize VCF files, we have to trust that the upstream steps to produce these VCF files, which Shaw mentioned previously, were done using established best practices. If this was not the case, it could certainly skew our results. And with that, I want to thank you all for listening, and we'll take any questions.